Hello, my name is Carol Eibel, and today we're going to examine the immune system, part 2, with emphasis on the innate immune system and human beings. Pathogens can cause devastation to humans. For example, the influenza epidemic in 1918 to 1919 killed 22 million people in 18 months. I can remember my grandmother telling me the story of how her father died in the prime of his life in 1918. Another example, 3 million people will die from malaria this year alone. Since 1980, over 619,000 people have died from AIDS in the United States. Worldwide, it is estimated that another 1.8 million will die in the next year. So the immune system is our defense system to fight against invading pathogens. The immune system has two parts. The innate immune system, which 5 years ago was called the nonspecific immune system. Heads up, on the AP exam you may see both terms. The innate immune system includes barrier defenses like your skin, mucous membranes, and their secretions. If bacteria should be able to get past your physical defenses, well then you will activate some internal defenses like phagocytic cells, antimicrobial proteins, the inflammatory response, and natural killer cells. In addition to that, you may also activate the adaptive immune system. Again, heads up, 5 years ago it was called the specific immune system, and a couple of years ago it was called the acquired immune system. Today, in the most recent textbooks, it is called the adaptive immune system. The adaptive immune system includes a cell-mediated response in which cells will kill infected cells, and the humoral response, involves the production of antibodies, which are probably molecules that you have heard of before. Alright, so in talking about the innate immunity, it is more rapid. The cells have receptors that recognize signature molecules that are shared by pathogens. It involves the activation of phagocytes. And as we've mentioned, it can also involve chemical responses that produce things like complementary proteins, antimicrobial proteins, and interferon. The adaptive immunity is much slower. It may take a week to 2 weeks before it's at its maximum effectiveness. It involves as we mentioned, two responses. The cell-mediated and the humoral response, which we will talk about later. So we are right now going to focus on the innate immunity. The first line of defense is the integument system, which includes your skin and mucous membranes. Well, your skin provides a physical barrier to the entry of pathogens. Your epidermal cells contain a protein called keratin. It's a structural protein that helps prevent pathogens from entering your body. And then you have those mucous membranes. Mucous membranes produce, no surprise, mucus, which help trap the pathogens before they can enter your body. Your skin also produces fatty acids, and some of those fatty acids can be toxic to certain pathogens. And you also have secretion from your tears, your sweat, and your oil glands that also have some toxins that can be detrimental to bacteria. Alright, so let's look at some ways pathogens can enter your body. Well, it is indeed possible that you can breathe pathogens into your body. There are airborne viruses and bacteria that you could just simply inhale. So they're headed for your lungs. However, your trachea are lined with ciliated cells, and there are other cells that produce mucus. The mucus will trap the pathogens, and then these ciliated cells will beat in unison to move those pathogens out of your trachea. Alright, so that's one way that pathogens can enter your body. A second way that they can enter your body is if you should ingest them. Take an apple. Even if you wash that apple for 5 minutes, there are still going to be lots of bacteria on that apple. So you eat the apple, and now the bacteria is in your body. It goes down the esophagus to your stomach. Your stomach actually produces hydrochloric acid and gastric juices. And those gastric juices with the acid has a pH of about 1 to 2. That will kill most of the pathogens. Alright, what about your urinary tract? That is also an opening to the outside where pathogens can gain entry. However, your urinary tract also has a lower pH. It's acidic. It's not conducive to certain bacteria, and your urinary tract is constantly being flushed with urine, and that will cause pathogens to be flushed out the body. Pathogens can also enter our body through our tear ducts. However, your tear ducts also produce tears, just like the urinary tract is going to flush out those ducts. In the tears, there's an enzyme called lysozyme. Lysozyme is an enzyme that reacts with the bacteria wall of pathogens, breaking those cell walls down. When that happens, osmosis will occur, and then those cells will burst open and be killed. Also, bacteria might enter your body through the reproductive tract. However, like many of your other ducts, has a lower pH and is not conducive to pathogens entering. But let's just say somehow, some way, there are some pathogens that turn around and make it past those physical barriers. What's going to happen is you are now going to your second line of defense in the innate immune system, and you're going to activate phagocytes. Phagocytes will undergo phagocytosis. And the question might be, where in the world do phagocytes come from? Well, phagocytes are a type of white blood cell, or we call them leukocytes. They are made in the red bone marrow of your bones from blood stem cells that undergo mitosis. They produce two types of cells, myeloid stem cells and lymphoid stem cells. The myeloid stem cells are responsible for making red blood cells, platelets, and then all of these white blood cells right here. Eosinophils, basophils, neutrophils, monocytes, and dendritic cells. And in addition to that, the lymphoid cells are responsible for producing lymphocytes, the B and T lymphocytes, and then another white blood cell called the natural killer cell. White blood cells are found circulating in your blood. However, we also find them in our connective tissue, the lining of organs, our lymph nodes, and also near the surface of our skin. So just a little bit about each of these different kinds of white blood cells. As I said, most of them are phagocytes, which means they can undergo phagocytosis. So neutrophils are one of the most numerous phagocytes. They're the first to arrive. You make approximately 1 billion every day. Right here is a picture of a neutrophil. However, they only live a few days, and they're what we call expendable cells. Eosinophils, you have fewer of those. They're weakly phagocytic cells that can destroy invaders that are clumped together. However, they have found out, this is kind of crazy, that they can work together to kill parasitic worms. Two other types of white blood cells include basophils and mast cells. Basophil cells are found circulating in your bloodstream. Your mast cells reside in your connective tissues. They are present in tissues characteristically surrounding blood vessels and nerves. They are especially prominent near boundaries between the outside world and your internal body, such as the skin, the mucosa of the lungs and digestive tract, as well as the mouth, nose, and your mouth and nose. Both mast cells and basophils, when damaged, will release a chemical called histamine, and we'll talk about histamine because it's a part of the inflammatory response. And then a fifth kind of white blood cell that you have in your body are called monocytes. So here is a picture of an immature monocyte. Not too impressive. However, when a monocyte arrives at the site of an infection, it will undergo a dramatic change. I like to think about it like Superman going into a phone booth and undergoing his changes. Da-da-da-da-da-da! They become a large macrophage involved in phagocytosis. So when they arrive at a site of infection, boy, do they start mopping up. Also, as they undergo phagocytosis, like right here we see a phagocyte engulfing this pathogen, it will break the pathogen down, and then it will take bits and pieces of that pathogen and put it on the surface of its cell membrane. And you think, well, why would it do that? It turns out that these macrophages now will be important in activating the adaptive immune response, and they become something called an antigen presenting cell. It's kind of like they are presenting trophies to certain types of T cells. Alright, so they always thought that macrophages were the only cells that were connecting the innate immune system to the adaptive immune system. However, recently they have found another connection. That other connection are the dendritic cells. And I like to think about dendritic cells as the king of the immune system. Dendritic cells are found in your skin, your nasal passages, your intestines, your spleen, things that are close to where pathogens will first enter the body. In terms of population numbers, they are smaller than other phagocytes. In terms of size, they are smaller than macrophages. However, they also undergo a dramatic change. Here is an immature dendritic cell, and then here is one that has undergone a change. You will notice these long, thin extensions. When they were first discovered, they reminded the scientist of dendrites on neurons, so they were called dendritic cells. Also, they have found that dendritic cells, like macrophages, can become those antigen-presenting cells. Dendritic cells were discovered by Dr. Ralph Steinman and Dr. Zanvil Khan. Steinman is the one who coined the term dendritic cells in 1973. He continued to investigate dendritic cells to determine their function. As I mentioned, it was just the macrophages that were thought to be the connection between the innate and adaptive immunity. However, people now believe dendritic cells are more important than the macrophages in becoming antigen-presenting cells. In 2011, Dr. Steinman was awarded the Nobel Prize in Physiology or Medicine for his contribution to the immune system. However, Dr. Steinman died 3 days prior to the announcement. Since the Nobel Prize is not awarded to people who have already passed away, it presented a problem to the committee. The committee, however, decided that to award the prize because it was made in good faith, and so therefore the decision would remain unchanged. So the question is, how do phagocytes work? How do phagocytes know when there's a pathogen and or when it's your own body cell, healthy cell? It's kind of like the story about the amoeba in that last PowerPoint. It should not surprise you, phagocytes have these receptor sites called toll-like receptors. They have the ability to recognize signature molecules that are unique to certain pathogens. For example, if there's a class of bacteria that have flagella, and they contain a protein called flagellin. Now, you will not find flagellin in any human body in any of our cells. So if this molecule should come around and attach to that toll-like receptor, that basically is notifying that cell that it has a pathogen. It may undergo phagocytosis and form a little vesicle and then inactivate the pathogen. In addition to that, this receptor site may activate some biochemical pathway that may cause an inflammatory response or the production of antimicrobial peptides, which will inactivate a particular pathogen. This is toll-like receptor number 4. It recognizes a certain lipopolysaccharide that's unique to a certain pathogen. This right here, this toll-like receptor number 3, recognizes double-stranded DNA, which is only found in certain viruses. So once the phagocyte arrives at the site of infection, it will start to do phagocytosis and start engulfing and cleaning up and bringing in all these pathogens. And unfortunately, phagocytes don't know when to stop. And so they can continue this process until they have so many pathogens inside their cell that they will burst open. So if you have an infection and you have that little kind of yellow-green oozy liquid oozing out of where the pathogen has gained entry, those are phagocytes or white blood cells that have given their life in defense of your body. I think that's kind of romantic. Alright, so now let's move on and talk about another white blood cell, but it's not a phagocyte, that helps in the innate immune system, and those are the natural killer cells. Well, I'm sorry, before we do that, just to review, this interaction of phagocytes with signature molecules is an example of cell signaling. So this would be flagellin, or that lipopolysaccharide, attaches to a receptor site. The receptor site activates a signal transduction pathway, and then there is some sort of response. This response may be the production of antimicrobial polypeptides to inactivate the pathogen, or it may activate the inflammatory response, but there's some sort of response that takes place when this pathogen attaches to that receptor site. Alright, so now back to our natural killer cells. Natural killer cells are not phagocytes. They are made by lymphoid stem cells and not myeloid stem cells. And what is unusual about these natural killer cells is that they can detect infected cells due to a virus or cancerous cells. When you have a cell that's infected by a virus or a cancerous cell, it causes changes in the plasma proteins. Natural killer cells can detect those changes. When they detect those changes in a cell, they will kill that cell by secreting chemicals into the infected cell, or puncture the cell membrane and causing the cell to lyse. So this is a natural killer cell that has detected a cancer cell and indeed has causing that cancer cell to lyse. Alright, so we have a movie coming up about natural killer cells.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrap of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
Wow, that was really impressive, huh? Well, just a second here. It's going to take me a second to get my pen back. Alright, so it also turns out that phagocytes can release certain chemicals to help in the innate immune response. And these chemicals can cause the inflammatory response or the production of those antimicrobial polypeptides. So one of those chemicals are chemokines or kinins, and they're released by certain phagocytes. And when they're at the site of infection, they're more concentrated at the site of infection, and then they diffuse outward. And so what happens is that creates a gradient, if you will, where here it's very concentrated. This is the site of infection, and here it's not so concentrated. So now that gives certain other phagocytes kind of like a map to where is the site of infection. So it draws them to the site of infection. Also, these chemicals will cause increased circulation and capillary permeability so that the surrounding tissue will have more liquids. In addition, some phagocytes will release complement proteins, and these complement proteins comprise about 30 proteins, and they can support the innate immune system several ways. Number one, these proteins have the ability to create pores in invading bacteria, causing water to rush in. So here's a bacteria cell. Here is a pore that's created by the complement proteins. You see it kind of like drilling in. And then once that pore is created, water comes rushing in and the cell lyses. Here is another movie we're going to watch.
Part of nonspecific immunity is a set of about 20 proteins called complement. One mode of action of complement proteins is to form a membrane attack complex when triggered by foreign cells or body cells infected by virus. Clusters of complement protein perforate the cell, whose contents leak out. Complement also stimulates phagocytic white blood cells to engulf these foreign or infected cells.
Alright, so much for the complement proteins, and I'm going to get my pen back here. Alright, here is another diagram showing you how these complement proteins kind of drill themselves into the membrane of a pathogen. Kind of cool. Alright, another thing complement proteins do is that along with certain antibodies, they will coat a bacterium. So here's a bacteria, okay, and here are these complement proteins. What happens is that phagocytes can actually detect these complement proteins and are attracted to the complement proteins. They will promote phagocytosis and the engulfing of that bacterium. And once phagocytosis takes place, the bacteria is destroyed. Da-da-da-dum! Alright, a third type of chemical produced by the innate immune system are interferons. Interferons are proteins made by a virus-infected cell. It's secreted and transported to neighboring cells to prevent viral infection from the infected cell. It reminds me of the systemic acquired resistance that certain plants exhibit, or called SARS. Alright, so here is an infected cell. Here the virus comes in. What happens now is that it activates certain proteins, which causes the production of interferon. Interferon travels to neighboring cells, attaches to a receptor site. Those receptor sites turn around and a transduction pathway takes place where now antiviral genes are activated, and so now certain proteins are made to prevent infection by other viruses. So this is now said to be in an antiviral state. So even though this poor cell is going to die, this poor cell produced a signal in the form of interferon to prevent that healthy cell from being attacked by the virus. The fourth type of chemical produced by the innate immune response are the histamine release and the inflammatory response. Remember I mentioned that mast cells and basophil cells, when they are damaged, they can release a chemical called histamine. Once histamine is released, what happens is that it causes here the capillary beds to become leaky. When that happens, fluids will move out of the capillary beds, Okay, they also will allow blood clotting elements to form. All right, these chemicals that have been released will cause the attraction of leukocytes, or as we would like to say, those leukocytes are phagocytes. They arrive at the site of infection. It may be a monocyte, and that monocyte, here we go, undergoes that transformation, becomes a phagocyte. They start doing phagocytosis and they start cleaning up and mopping up. Meanwhile, the blood clotting elements have formed blood clots, and here's a scab. And so now the fluids are returning back to the capillary bed. The redness will go away. The swelling will go down. An interesting chemical that also is released is one that causes the nerve endings to become more sensitive, so that your body realizes that this area has been injured, so that you don't continue to use that part of your body and causing further damage to that area. And that is called the inflammatory response. Here's an actual picture of the inflammatory response. The inflammation will continue as long as the pathogens are present. But remember, the phagocytes are there. They're starting to clean up the pathogens, phagocytosis, getting rid of the mess. When the phagocytes complete their job, what happens is that the macrophages will begin to secrete substances that will suppress the inflammatory response and promote tissue repair. So this is a case of homeostasis and making sure that the inflammatory response does not get out of control. Okay, if for some reason the stimulus if for some reason the stimulus stays there and the inflammation continues, what will happen is you will get a chronic inflammation, and that can cause diseases such as asthma, Crohn's disease, rheumatoid arthritis, arterial sclerosis, diabetes, and cancer. So it's important to make sure, or the body, to try to remove that stimulus and get it out of the way. Okay. Inflammation can also cause a fever. The question is, are fevers good or are they bad? Some cytokines that are produced will stimulate the brain to make prostaglandins. These prostaglandins will stimulate the hypothalamus to set a new temperature set point. Alright, so the hypothalamus is in control of your temperature. So the prostaglandins are basically telling the hypothalamus, hey, you need to go ahead and allow for a higher temperature. Alright, so what happens is that in order to make your temperature go up, it will constrict the blood vessels in your skin, so that now the blood is more towards the core of your body. It will contract your skeletal muscles. So if you have a fever, you know how you shiver? Well, the question is, why are you shivering? That shivering causes the muscles moving back and forth, produces heat. That heat can be used to facilitate a fever. And also it causes the heart rate and your respiration to go up. Okay, so a fever then will increase circulation, increase enzyme activity, and metabolism. However, some pathogens will reproduce slower at a higher temperature. So all those things combined will help fight an infection. However, if the temperature is too high, that can also be bad. You do not want a fever to go beyond 105 degrees, because if it goes beyond 105 degrees, you can turn around and you can you can have some serious damage to your proteins because they're going to undergo a conformational change and so forth. So it is not good to have a temperature 105 degrees. However, if your temperature is going too high, your body will respond by sweating, opening up the blood vessels in the skin, and slowing down your metabolism. Alright, so we have looked at the innate immune system today. I hope you've enjoyed it, learned lots of stuff, and that you'll be able to use it on the AP exam. Have a good day, folks.